Okay, good morning. Make sure I'm sharing correctly. This is Dr. Garais, BIO117. This is our last lecture together. I can even hear the tears rolling down your cumulative faces. All kidding aside, let's get this show on the road. This is chapter 24, and we're going to talk about the lovely topic of genetics. And also I will have also today as well. Sorry, I'm a little bit distracted. I'm in the room and I smell burning. I hope that's not me. Okay. School's on fire. Hopefully someone will inform me. Again, we could fast forward this part. What are we studying? The genetics, which is inheritance, the characteristics. And remember, the genes is the DNA sequence, or the genotype is the code. And genome is the complete set of genetic instructions. So it's everything. And that's what your genes has. It has everything. The protein encoding genes, that's your exome, right? And this is also the one chapter where we will be discussing and you will be responsible for pathologies. So cystic fibrosis, I want you to know that it is, of course, a genetic disorder, and it has defective chloride ion channels, which results in thick mucus, and it gives, it gums up the pancreas and the lungs for these patients. We could see here that, right, from really small to even, to relatively bigger, but it's still the nucleus of the cell, which has the DNA, and it's very small. And the DNA, because of the hydrogen bonds that are located in between the base pairs, adenine, thymine, guanine, and cytosine, that forms this double helix structure, and that gets supercoiled, and then around these balls right here of protein called histones, Histones, then it coils, and then into what we all know and love, this little butterfly thing, well, I call it a butterfly, and that is your chromosome. And we have 23 of them times 2, which is 46 to make a complete human being. Of course, we already know DNA gets copied and then transcribes it onto mRNA. The mRNA then gives it over to transfer RNA, and then it translates that. But in cystic fibrosis, it's a genetic problem, part of the DNA code is messed up. So it's going to gum up your sinuses, your airways, particularly your pancreas, and the bile duct, and your intestines. So too much of a good thing, which is mucus, becomes a significant problem for this patient. The neat thing about inheritance or the transfer of the genetic code into some sort of visual or some sort of observable characteristic is that genetics you can start predicting things. And the way geneticists and physicians start predicting things is through understanding and knowing the modes of inheritance. And we're going to talk about the modes of inheritance shortly. Karyotype. What's a karyotype? A karyotype is a chart displaying all 23 chromosomes, and I'm going to show you a picture of that in a moment. Chromosomes 1 through 22 are autosomal, and chromosome 23 is your bar body, also known as your sex chromosome. And we already know the 23rd pair of chromosomes, of course, one from daddy, one from mommy. If you have XX, you're female. XY, you are male. Okay. Now, what are alleles? Alleles are they're 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 the pair of the code that we talked about in lecture. The capital A, capital A, capital A, lowercase a, and lowercase a. So those are the alleles because things happen in pairs in genetics, and it's because, of course, right, we have chromosomes. So if they're homozygous, like homozygous dominant, capital A, the two pairs of alleles are identical. If they're homozygous recessive, that's lowercase a, the two pairs are identical, but they're what? Lowercase a, that means it's a recessive trait, not a dominant trait. A recessive trait is something that most likely won't show up unless it's alone, like in homozygous recessive. But a dominant trait usually is what it's called, dominance, so it will most likely show up. Or a person could be heterozygous. And remember, capital A, lowercase a is a pair of alleles that are heterozygous. Hetero meaning different. And we already know that hetero from the lecture is heterozygous is could also be a carrier. Here's a picture of a human karyotype. You can see there are pairs of chromosomes. We have 23 of them, 1 through 22 autosomal, and the 23rd is your bar body, which is your sex chromosome. We already talked about what is dominant and what is recessive. Dominant means it'll show up, right, in the real world. Recessive, it may show up, but is most likely masked by the dominant gene. When there's no dominant gene present, like a capital A, then what will we see? We'll see the recessive trait. So genotype, think what? The letters, the code, the pairs of alleles. The phenotype is the appearance or trait, what's going to come out. Now, all of this is based on Mendelian inheritance of the dominant and the concept of dominant and recessive genotypes, which then show dominant and recessive phenotypes. We will go over several examples of the Punnett square, and that's the table with the 4 boxes. It has mommy's alleles to the left and daddy's alleles on the top. And I said in class they can be interchangeable. Mathematically, yes, but now that I'm looking at this, there's rules to it. Mommy is always on the left vertical side, and daddy is always on the top part of the box on the horizontal side. And what's the pedigree? That's the squares and circles with a diagram showing the family relationships. So it shows not only the genotype and the phenotype of each member. Circles always represent females, and squares, males. Shaded areas are colored areas, and they usually, in pedigrees, usually have some sort of key, shows the affected people. Half shaded areas show carriers, while if there's no color or unshaded, that means that person is perfectly normal with respect to disease and has no trait and has no disease. A classic example is skin pigmentation. So let's look at this chart right here. So of course, daddy is always going to be up top because you see that sperm cells. Mommy is always going to be here. So let's say daddy was heterozygous dominant, has a capital A and a lowercase a. Mommy was a heterozygous as well, capital A, lowercase a. So they're both carriers because they're heterozygous dominant. And what the Punnett square does for us is it then does what? Susses out all the possible combinations. And they can be represented mathematically as a ratio or a fraction. Okay? And if you have some issues with math, ratio, and fraction, come see me. Don't forget that I also used to teach and tutor math. So let me see if I can review this. So this is mommy. end. And, okay, so of course here, this horizontal part, this is of course mommy, mom. Oh, cool, it works. I did this. And of course, up here, father, dad. And the dad will always be in the horizontal, mom will always be in the vertical. So if we look at these combinations, this A goes here, because it's capital A. This capital A from dad goes here. So it forms in this box, capital A, capital A. This capital A, dad, of course, goes down here because that matches up to this lane. And the lowercase a goes here. And you do that for all 4 boxes. This little a. And typically when we draw out the pair of alleles, which is this is a pair, right? And the code, we always put the dominant one first. So even though this one, right, came A, right, like we did mommy first, well, the way we write it is capital A lowercase a. So you can see here that 1 out of 4, or 25%, right, 25% here. It's perfectly normal. Okay, so the skin pigmentation, they're not going to have a problem. Now, the heterozygous dominant, these two, they're both carriers. So there's 2 of them. So 1/2, 1 divided by 2 is what? 50%. 50 over 100, 50%. So I could ask, for this particular Punnett square for albinism, 1/4 of the kids not going to have any problem. 50% of the children are going to be what? Carriers, but they'll appear normal. Their skin will be fine. But 25% of the kids, the chance of a child to have albinism, right? No melanocytes is 25%. 1 out of 4. Okay. So potential questions I could ask, right? If I show you this Punnett square, you'll tell me this is daddy's genotype, capital A, lowercase a. Phenotypically, he's normal because for skin pigmentation, for albinism, a carrier doesn't have the disease. Mommy is also a carrier, but 1/4 or 25% of their offspring is going to be perfectly normal, no issues, and no genotype because it's what? Capital A, capital A. But 25% of them will have the disease, lowercase a, lowercase a. That's in albinism. So that is an example of it's an example of Mendelian genetics and a Punnett square. Sorry, I'm distracted. I should have closed my door. Let's talk about other kinds of diseases. Now, another kind of disease is an autosomal recessive disorder. And we already know cystic fibrosis of Heterogeneity. So if so there's of course 3 combinations. And cystic fibrosis, just like albinism, has an autosomal recessive disorder. So who gets affected? Right, the homozygous recessive, or the lowercase. So this person has the disease of cystic fibrosis. And of course, 25%. Perfectly normal. 25% going to have the disease. 25% of the kids. And 50% will be, of course, unaffected carriers. So lump cystic fibrosis and and cystic fibrosis and albinism together because they are examples of autosomal recessive. And I'm going a little bit crazy on the mark. That's right. Like a kid with a new toy. Now, what is an autosomal dominant? Now, before we were talking about an autosomal recessive, the carriers don't get affected. But in the case of an autosomal dominant, as long as there's at least one capital, right, which here, then someone's going to get the disease. Okay, so this typically would be a carrier. You have a capital and then a lowercase. It's a heterozygous dominant. Well, in an autosomal dominant, right? As long as we have that one mutant allele, which is this one, we're going to get it. Okay. Next one. So Huntington's disease, think what? Heterozygous, they're going to get it because it has one lowercase. So in Huntington's, it goes in Huntington's, you have what? You got a 50% chance of getting it if one of your parents is a carrier. Now let's look at this. This is a pedigree here on the bottom of what you see here. Now Dan, the daddy, Anne is the mommy, and then they had two kids, Pam and Eric. Now if we look at this, I could ask you questions like this. Who's dad and who's the mom? Of course, you see this. This is Gen 1 or first generation. So you'll say Dan is the father. Anne is the mother. Who got affected? This is the pedigree for Huntington's. And you'll say who? You'll say, well, it's like what? Dan, the dad. Who's unaffected? Mom, Anne. They had two kids. What are the odds that one of the kids? What are the odds that the kids are going to get the disease? Well, it's 50%. And what did it actually happen? In the next generation, which were the kids, Pam is the daughter of Dan and Anne. Eric is the son of Dan and Anne. Who got hit? Eric. So who has Huntington's? Dan, the father, and Eric, the son. And that's the kind of question that I'll ask on an exam if I show a pedigree. Let's look at this one. Regarding cystic fibrosis. Now remember, cystic fibrosis, a carrier doesn't get hit, but let's ask some basic questions. Who's the dad? Gen 1, dad's Joe. Who's mom? Mary. Now you notice they're only colored halfway. If they're colored halfway, that means they're heterozygous. That means, and for cystic fibrosis and albinism, they're carriers. And carriers are perfectly normal. So Joe and Mary, they're okay. And for each child, there's a chance of what? 25% being perfectly normal, 50% being carriers, and 25% being affected. But let's see what happened in real life. And that's what happens here in this pedigree. So I could ask, how many children did they have? You look at second gen, Joe and Mary had 3 children. Is Bill sick? Nope. Bill is perfectly normal. Is Sue sick? Nope. Sue isn't sick. She doesn't have cystic fibrosis. But guess what? She's a carrier. Is Tina sick? You'll notice that Tina is colored what? Completely. Therefore, Tina is sick. Therefore, Tina is homozygous recessive, right? Lowercase, 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 lowercase. So this is an example of Tina. So again, to reiterate, Joe and Mary are the parents. Joe and Mary are carriers. They had 3 children. Bill, unaffected. Sue became a carrier and is, of course, normal phenotypically because cystic fibrosis and albinism is autosomal recessive. So who got hit? Tina got the disease because she has what? Both pairs of alleles are recessive They're both lowercase, therefore she's the one with the disease. So those were the examples of Mendelian inheritance, but they're not the only ones examples. There are other types of examples of Mendelian inheritance, and they are codominance, incomplete dominance, multiple alleles, pleiotropy, and sex-linked inheritance, and you have to know all of them. So let's look at what each one is. I want you to think codominance, that's when the heterozygous, both alleles in the heterozygote gets expressed. So that means what? Like an ABO. You can have an A and a B, and both will get expressed because they are codominant. So think ABO blood typing and codominance, and think what? A and B alleles. Now, if you don't have any A or B alleles, and goes, what will you have? You'll have what? Recessive progeny. And remember, O doesn't have any A or B glycoprotein. Multiple alleles, inheritance in which there are more than two alleles. Again, another example is what? ABO. Okay, so think codominance, ABO blood type. Multiple alleles, ABO blood type. Incomplete dominance. Incomplete dominance, I want you to think of sickle cell anemia. And of course, we've already talked about sickle cell anemia. So the homozygous, capital S, their genotype is normal and their phenotype is also normal. But it is the homozygous recessive SS genotype that will have sickle cell anemia. That person will have severe symptomology. But the heterozygous, which will be typically a carrier, has sickle cell trait. They have a mild form of sickle cell anemia. Okay. Another example of incomplete dominance is familial hypercholesterolemia, because there are levels. So a homozygous recessive person has what? A lot of problems. But the heterozygous has what? Some LDL problems. That's incomplete dominance, hypercholesterolemia. Nice chart. Let's get the pleiotropy. Polygenic inheritance and pleiotropy. So what are polygenic traits? There are traits determined by more than one gene. And classic examples are height, skin color, and eye color. They're multifactorial. So think polygenic, multipoly or many genes. Pleo, many or different. Tropy is growth, so different growth traits. So think of what? Height, skin color, eye color, pleiotropy. Another example of pleiotropy, but it is actually an autosomal dominant defect. Think Marfan's. And remember, I talked about how Jim Carrey, I felt that, his face and his fingers and his body stretches a lot. And you can see the polygenic traits. There's multiple traits for different skin tone and shades. And it's the same thing for your eyes, and it's also the same thing for your height. The majority of people you could see will be on the standard deviation of what? Somewhere in the middle. Again, another polygenic pleiotropy, right? If you have absolutely recessive, all recessive genes, you'll be devoid of any color, which is the default is blue, or very light blue eyes. But if you have all the dominant colors, right? We all know what happens when you take all the paints and you mix them. You get what? A very dark color. You get black eyes or really dark brown eyes. Chromosomal inheritance or sex-linked. Remember, it's usually X chromosome. It's usually from mommy, right? That we have sex-linked traits. So if it's sex-linked, that means we could track it through the sex of the child. So if the sex of the child will actually be the predictor. And remember, regarding on male versus female, next slide will show us that the odds of having a male child and a female child is what? Perfectly even, Steven. 50%, 50%. So I could ask on an exam, right? You got, of course, daddy up here, mommy up here. What are the odds of the baby being female? 50%. What are the odds of the baby being male? 50%. So it's what? It's a toss-up. Now, remember, X-linked traits are way more common connected to mommy. Such examples are red-green color blindness, hemophilia, and Duchenne's muscular dystrophy. So if I mention any of those, that's will be what? Sex-linked and linked to the X chromosome, linked to mommy. Now, the Y chromosome is much more rare and not worth clinically talking about. Okay, what are the factors that affect expression of genes? So penetrance is how much of the genotype will actually get expressed, right? Which degree of expressivity in the phenotype. So complete penetrance means that the disease-causing allele will definitely do what? Express in the real world as a physical trait or physical defect. Incomplete penetrance, right? Majority of the traits goes, you inherit some of the allele, not all of it. And then variable expressivity means there'll be different types of phenotypes and different types of intensities in different people. Nice to know, but penetrance and expressivity, knowing the different shades of this, nice to know, but penetrance and expressivity, it's better to know. Heterogeneity. Okay. Now, hetero means different genes. So we already talked about that in a way where different genes encode for different things, and there's more than one gene that encodes for it. And so blood disorders or blood dyscrasias are classic example. And hemophilia, we already talked about that. Sex linked, we already talked about. Sex influence. Regarding sex influence, and the classic example of baldness is dominant in males because the main reason of baldness is excessive testosterone. So when you're balding, it's because most likely there's an excess of testosterone. Multifactorial, we already talked about that in polygenic or pleiotropic genes. It's eye color, height, skin color. This is classic. You can see how it was mentioned several times in this lecture. Therefore, what are the odds of it coming out? Chromosomal disorders. So when in meiosis, especially in meiosis 2, when the chromosomes rearrange, they can rearrange in the wrong way. And it could be inverted. It could be exchanges that went wrong. Or it could be they didn't exchange. Like some of them, like in the trisomy, we're going to talk about in a minute, they don't separate like they're supposed to during the anaphase part of meiosis 1 or 2. Polyploidy, we already talk about that. Let's talk about this. Aneuploid. Cells that have a normal number of chromosomes, euploid. Because what's normal? The prefix EU. That means it's normal. And the prefix "and" means what? something's missing. And remember the terms haploid and diploid. That means we're talking about number of chromosomes. So if my patient has 46 chromosomes, we're good. My patient is euploid, right? But if my patient's missing something, most likely because it didn't properly separate during anaphase of probably meiosis 2, well, aneuploid, and there could be a meiosis error, and it's called non-disjunction. And then non-disjunction leads to things like Down syndrome, which is the trisomy 21. So chromosome 21 was supposed to split, like double up and split up, but for Down syndrome, right, it is doesn't separate. So you'll have autosomal aneuploidy, something's going to be missing. So Down syndrome is trisomy 21, Patau is 13, Edwards is 18. Must know is Down syndrome, trisomy 21. Just know that Patau and Edwards are quite severe, and the baby usually doesn't live. Oh, the sex chromosome, I forgot to mention aneuploidy. Things like XO. So this particular female patient doesn't have an XX, it's missing something. So they're going to have delayed or diminished sexual development. You know, the breast won't be fully developed, fat distribution won't be there, you know. And a classic example is, who's the actress? What's her name? She was in the movie Halloween. Jamie Lee Curtis. If you look at Jamie Lee Curtis, right, she has classic Turner syndrome. Breast development, Tanner stages isn't too developed. If you look at the shape of her body, it's very straight. It's not as curvy as somebody who would have the complete XX chromosome. And you can see it's you can see the survival rates pretty poor for Patau and Edwards syndromes. Downs, you know, the majority of them will live, but again, it's a trisomy. It is a chromosomal defect. They don't life expectancy for Down syndrome patient, they don't live as long as the patient who didn't have trisomy. Some prenatal tests, nice to know, not really too crucial, because there's so many other things that I could ask you about. And that's ultrasound, maternal serum markers, we get that in the maternal blood, cell-free fetal DNA, again, maternal blood. I could do a chorionic villus sampling or CVS. That samples the outer layer or the outer membrane of the fetal sac, and the, of course, amniocentesis that goes right in, and that is ultrasound guided. And here's a picture of it. And this one's like ultrasound guided here. And this is the transducer. Make sure we don't hit baby. Make sure we get through all of it. 3D ultrasound, nice, but not necessary. It's just nice to look at your kid like, ooh, look, it looks like. But the ultrasound is used for other things. We also want to see all the organogenesis. We also count the somites. Another thing we also count is the fluid, the amniotic fluid. Because too much amniotic fluid, not a good thing. Too little amniotic fluid, not a good thing. Genomics and healthcare, nice to know, detection of Mendelian disease. So that wraps it up. Much. And I'll see you in class, and let me get some sample questions out to everybody today. Have a good one, everybody. Bye now.